Today we're going to discuss PCR and the gene of interest, that is 16S or RNA. PCR itself is used to amplify a particular region or segment of DNA, producing enough DNA copies to be analyzed using other techniques like electrophoresis, for example. To prepare the PCR, we will combine sterile water, 27F, which is forward primer, 1492R, which is reverse primer, PCR bead, and the DNA sample in all 4 tubes for the PCR process. The water and DNA will vary in each tube to make a total volume of 25 microliters. More DNA, less water, etc. It is put in the thermocycle for about 2.5 hours. We will denature it in the thermocycler at 95 to 96 degrees, but the DNA itself won't be denatured because the TAC is a thermostable DNA polymerase. It will automate the repetitive step of amplifying specific DNA sequences. This step will allow the DNA to come apart so the primers can find their complementary strand. In the annealing step, the primers will sit and anneal to the DNA. The temp depends on the sequence of primers. We then cool the reaction so the primers can bind to the complementary sequence regions. The last step includes raising the temp to about 72 degrees so the TAC can extend the primers and synthesize the new strand of DNA. The cycle will extend 20 to 40 times, and that is PCR.